All right, let's get started, shall we? So, good morning in Seattle, good afternoon in New York, and good evening in Germany and the UK. And welcome all of you to the fifth webinar of Therapeutical cycle of webinars. The webinar today is entitled Neuroscience Frontier Challenges for 2020. First and foremost, accept my apologies as the organizer of this webinar, Raúl Delgado, will not be presenting this neuro webinar. Unfortunately for us, he will not be with us today because he has recently become a father and so he needs to perform very well his new responsibility of taking care of both the mother and the baby. So my name is Enrique Gallego and I will be presenting this neuro webinar for you today, as well as guide you through all the different talks. May I remind you to all the attendees that if you wish to make any questions, you could do so by writing it down in the question section, as well as by raising your yellow hand. Here it is, located in the menu column. All the questions will be answered at the end of each block, each talk. So let's kick off this neuro webinar by presenting our first speaker for today, Dr. Ana Maria Cuervo. Dr. Cuervo is professor of developmental and molecular biology, and she currently holds the Robert and Rini Belfer Chair for the study of neurodegenerative diseases at the Albert Einstein College of Medicine. She is also the co-director of the Einstein Institute for Aging Studies in New York, USA. Dr. Cuervo's group is interested in understanding how altered proteins can be eliminated from the cells through the lysosomal system, autophagy, and how malfunctions of autophagy in aging is linked to neurodegenerative diseases. So, Dr. Cuervo, thank you very much for this opportunity, and please, I'll give you the word to start your seminar right now. Just give me 1 second. Ana Maria, make presenter. Okay. We can hear you now, yes.
Can you see the presentation?
We do, yes, we do.
Okay, terrific. So should I start?
Yes, please.
Okay, so well, thank you so much for having me here. I'm gonna be telling you a little about the interest of our lab that is mostly on the process of autophagy. So most of you probably already know what autophagy means, but it's just this process by which intracellular components of any kind, proteins, organelles, or even pathogens, when they reach the cytosol, are delivered to lysosomes for degradation. We are very interested in how this delivery occurs because that gave rise to different types of autophagy. For example, you can just sequester part of the cytosolic material in this double membrane vesicle or autophagosome that then fuse with the lysosomes in this process known as macroautophagy. But lysosomes can also invaginate their membrane to trap little bites of the cytosol, and a very similar process occurs in the late endosomes. So these processes are known as microautophagy or endosomal microautophagy, depending on the compartment. And then in mammalian cells, there is a third way to transport proteins inside lysosomes, by which proteins are recognized by a specific cytosolic chaperone that deliver them to a lysosomal transporter, and this process is known as chaperone-mediated autophagy. Independently of these types of autophagy, we are mostly interested in 4 functions of this process in our group. In one hand, we are interested on how autophagy can be a very efficient mechanism to maintain the energetic balance of your cells because it allows for the recycling of amino acids or free fatty acids of the materials that are being degraded. We're also interested on the role of autophagy as a mechanism of cell defense because, as I mentioned, can eliminate pathogens from your cells. Because some of these autophagy processes are very selective, they can also contribute to the remodeling of the proteome and accommodating your cells to different conditions. But for the purpose of this talk, I'm going to be mostly focusing on the role of autophagy in quality control, the mechanism by which they remove abnormal proteins or organelles to maintain cellular homeostasis. So I'm just going to start with this process of macroautophagy, just giving you a couple of general ideas about the mechanism of this process. So in macroautophagy, as I mentioned, is all about sequestering materials in these double membrane vesicles or autophagosomes and then fuse them with the lysosomes. And you can basically recapitulate each of these steps by electron microscopy. You can see on the left cells in which autophagy is very active, and you can see how materials are being degraded in those vesicles. You can see before degradation this type of double membrane vesicles with material that is very similar density to the one in the cytosol. And then once lysosomes fuse, you start seeing this kind of profiles where you see degradation as clear areas, materials that you cannot recognize so well, and then finally you get the secondary lysosomes. During these recent years, there has been a lot of interest in trying to characterize the molecular players of this process. And there are about 35 genes that participate in macroautophagy, and they group in functional complexes. Just to give you some examples, you have this kinase complex in which the most important component is this lipid kinase, this VPS34, that is going to phosphorylate lipids in the surface of the organelles where autophagosomes are going to be generated. These are under the control of many different negative regulators. The best characterized is mTOR, that, as you know, is a nutrient sensor that normally repress the autophagy process. Once you don't have nutrients, mTOR gets activated, it gets inactivated, and then these kind of components that are repressed go to the site of formation of the autophagosome. You can create an elongation membrane just by building blocks that originate from the conjugation of a protein, this LC3, to a lipid, and two proteins, ATG12 and 5. And basically, this is making just grow in the membrane to trap the materials that you eventually want to send for degradation to lysosomes. This process can be non-selective or in bulk. For example, in response to starvation, the cell just degrades whatever it has around. But it can also be very selectively targeting specific components of the cytosol and giving rise to these terminologies such as lipophagy, mitophagy, depending basically on what you are degrading. We are interested in this process from the point of view of neurodegeneration in two ways. In one way, we want to know how autophagy contributes to the removal of pathogenic proteins, those ones that misbehave in many of these neurodegenerative disorders. And in the other hand, we are very interested how these pathogenic proteins can primarily alter the functioning of the autophagy process. So just to give you an idea of some of the studies, it is very clear that autophagy contributes to the removal of abnormal proteins, and this was first shown in the central nervous system by the labs of Mishima and Tanaka, that showed that if you eliminate autophagy, for example, selectively in neurons, you get a full phenotype of neurodegeneration with accumulation of aggregates and abnormal proteins, suggesting that you normally need autophagy in your neurons to maintain neuronal homeostasis. It's particularly of interest the removal not only of soluble proteins that could happen for almost every pathway, but in particular of proteins that are aggregates, because macroautophagy seems to be the almost only pathway that can really take care of these aggregates. And there is a lot of interest trying to figure out how these aggregates are being recognized by the autophagy machinery, for example, by ubiquitin and cargo recognizing proteins, and how then they are sequestered, are sent to degradation. From the point of view of neurodegeneration, there are many different ways in which autophagy can contribute to disease, because it seems to be a favored target for some of these pathogenic proteins. So in the following cartoon, I just summarize the different kind of diseases or neurodegenerative diseases in which alterations in each of these steps have been shown. For example, there are conditions in which pathogenic proteins can co-aggregate with autophagy-related proteins, and this contribute to decrease autophagy activity, for example, in Alzheimer's. Also, it has been shown that an increase in negative signaling, for example, through the insulin or mTOR pathway, can also decrease autophagy. And under those conditions, you really want to enhance the autophagy activity in order to resolve the problem. However, there are other conditions in which initiation of the process is okay, and the problem is in the recognition of cargo. For example, there are pathologies in which some aggregates can be nicely recognized as the one that you see on the right side. You see the aggregate, and then you see all these autophagy vacuoles around. Meanwhile, you have other aggregates of similar composition that seems to be escaping the autophagy surveillance. In other cases, the problem is not the cargo, the aggregates themselves, but the machinery that recognize the cargo. As I mentioned, you have to form these limiting membranes of autophagosomes, and proteins such as mutant huntingtin, the protein that is mutated in Huntington, seems to interact with the inner part of these forming membranes and prevent selective recognition, for example, of mitochondrial lipid droplets. So in these cases, if you stimulate autophagosome formation, this is going to enhance autophagy clearance, even if it's randomly. But ultimately, our goal is really trying to figure out why some aggregates are recognized and some are not, and also how to eliminate these pathogenic proteins that can be altering the autophagy recognition. Following with the scheme, sometimes the problem is later on in the process. For example, any condition in which you have alterations of the cytoskeleton might interfere with autophagy, since these autophagosomes move through the cytoskeleton through microtubules, or even the presence of proteins associated in non-specific manner to the surface of these vesicles can prevent the fusion with the lysosome. And then in the last step, at the end of the day, the one who does all the job is really the lysosome. So any condition in which you have compromise of lysosomal function also will result in accumulation of these autophagy vacuoles in the cytosol. And just to give you some example, I'm showing here in the left an electron microscopy of a brain from an Alzheimer patient, in which you can see a dilated neurite completely full packed with these autophagy vacuoles. So in this case, if you induce formation of autophagosomes, for example with rapamycin, you will just make things worse, because you will end with many more of those vesicles. So in these cases, the idea is to enhance the functioning of the lysosomal system in order to favor clearance, and not just to enhance the formation. So part of our lab is trying to figure out mechanisms by which you can enhance lysosomal function as a whole. And then just in the last 2 minutes, I just want to tell you about the other pathway, this chaperone-mediated autophagy, that also seems to be victim sometimes of pathogenic proteins in neurodegeneration. This process is very different, and all the activity occurs in the lysosomes. You don't form autophagosomes. The proteins degraded by this pathway are cytosolic proteins with a targeting motif that is recognized by a chaperone that delivers to a receptor at the lysosomal membrane, this LAM2A. So basically, whenever you need to eliminate the protein, it's recognized by the chaperone. The chaperone brings it to the surface of the lysosomes. This single transmembrane protein now multimerized, and you form this translocation channel that allowed for degradation of the substrates in the lumen. Through the years, and with collaboration with many terrific neuroscientists, we have identified that many of the pathogenic proteins that accumulate in conditions such as Parkinson, tauopathies, or Huntington contain targeting motif for this pathway. And in most conditions, the wild-type proteins, the ones that are normal, can be degraded by the proteasome, chaperone-mediated autophagy, macroautophagy, by different pathways. However, when you have the pathogenic variants, it is very common that these proteins are delivered to the surface of the lysosomes, but they don't translocate. They basically get stuck in the membrane, so then they will accumulate in the cytosol, and also because they just don't translocate into the lumen, they prevent the degradation of other substrates that are normally degraded by this pathway, further contributing to altered homeostasis. So we've been trying to figure out what is the problem with the translocation, and we got some ideas with our recent work with LAR2. LAR2 is the protein that is most commonly mutated in familial forms of Parkinson's disease, and as you can see here, contains many of these targeting motifs to the lysosomal system. And what we found is that while the normal protein is targeted to lysosomes, binds to the receptor, multimerized, and allow for degradation, when you have the mutant protein, it completely disrupts the formation of this multimeric complex. You can see here blue native electrophoresis showing the monomer of this receptor and the multimer as the 700 kilodalton protein. And once you add the mutant protein, the mutant LAR, this complex is disrupted. But even if you are the wild type at high levels, you can also get the disruption. And if you isolate lysosomes from animals that have altered expression of this LAR protein, you can see how this multimeric translocation system at the lysosomes is altered. So now that we know the defect, part of our effort is trying to identify or generate molecules that can enhance the formation of this multimeric complex and guarantee trafficking even in the presence of this pathogenic protein. Because our model is that once you have this disruption of this translocation complex, proteins such as alpha-synuclein and other proteins related with Parkinson will bind to the receptor in the surface of the lysosomes, but because they cannot translocate, they will oligomerize and will disrupt trafficking. And we think that this will further contribute to disrupt the system. So just in the last slide, I just want to summarize that I try to convey how autophagy can defend you against the proteotoxicity by eliminating pathogenic proteins, but how come in many of the pathogenic diseases or many of the neurodegenerative diseases, these systems, these forms of autophagy are also the victims of the pathology. So that's all for my comments. I think I went a little over time.
Yes, but it's okay.
Hello?
Yeah, can you hear me?
Yes, yes.
So thank you very much for this very thrilling and interesting seminar. And now we have allocated 5 minutes for questions. Therefore, if there is any question, could you please raise your yellow hand that you will find on your menu bar. In the meantime, because people usually is undecisive trying to make questions, I was thinking, I'm on the cardiovascular field, and I was wondering if is there any way to visualize this by, for instance, MRI or imaging this kind of diseases, and then correlate this to problems in autophagy, sorry.
So there is a lot of interest in trying to identify this process in vivo. But so far, the field has heavily depended on electron microscopy, as you saw through the presentation, that of course is just giving you a steady state, so it doesn't give you much dynamic. With the time, different labs, including ours, have developed animals that express fluorescent probes that are substrate for one pathway or another. So now you can visualize it using 2-photon microscopy through these probes. But of course, you need the animals expressing these probes. The field is trying to analyze some byproducts of this degradation that can be tracked by mass spectrometry or some of these techniques. But so far, unfortunately, it's not possible to differentiate what is the system producing these byproducts. So at the moment, it's all really in imaging.
All right. Thank you very much. We do have another question, and I'll give you the voice, Carmen.
I don't seem to hear. Yes?
Can you hear me?
Yes.
Okay. Thank you very much for this presentation. It was really nice. And the question I have is whether these processes are activity regulated.
Yeah, so you mean exercise?
Yeah, whether there is any neuronal signaling, for example.
Oh, yeah. So that's a very good question, because I mean at the cellular level, it is very clear that pathways like mTOR, Wnt signaling, sonic hedgehog, they modulate this pathway. But now there is a growing interest to see if firing of neurons can also activate this process. And apparently there are very nice examples now showing that increased activity can stimulate this process in theory, because you can also contribute to the recycling and degradation of some of the neurotransmitters. But the opposite is true. It's like when you disrupt the autophagy process, there has been very nice connections with epilepsy. So apparently you start having this regulation of firing in some neurons, and it seems to be related more with not accumulation of garbage, but problems with the energetic balance of the cells. I mean, this is a good mechanism to provide ATP, so that this regulation of this process seems to result in altered signaling at the energetic level.
Okay, thank you. And I have one more question. So it was recently shown that when mice sleep, there is a faster clearing of the kind of bad proteins or bad like beta peptides and etc. So I was wondering whether the autophagy process is also more active when the people are sleeping.
I missed the last part. When people are?
like sleeping or
Oh, it's
relaxed?
sleeping. Oh yeah. Yeah, no, that's a good point. I mean, in a way, it will activate this process because the process is done regulated by nutrients. So since
Okay.
when you are sleeping, you are not eating, and in
Yeah.
theory, you activate autophagy at night. I mean, you activate
Okay. So
autophagy
it
in
makes
between,
sense.
yeah, you activate autophagy in between meals. But the
Mm-hmm.
idea is that because we all snack so much, each time we activate autophagy less during the day. So the only period that you really don't have nutrients coming has become the sleeping time. So that's a good point, yeah.
Okay, thank you.
Hello. I do have another. Whoa, there's three more questions coming. I'm not sure if we should leave this at the end of the webinar, but let me ask you another question. Susanna is asking me to you if are there other cell-based assays to look at autophagy instead of endpoint staining?
Oh yeah. So there are very good reporters, and actually I'm happy to share them with whoever wants. Just drop me an email. So I mean, you can do a Western blot depending on what the kind of cells that you have and the amount of material. But it's a Western blot in which you inhibit the lysosomal process for 2 hours. So you basically have an idea how much is degraded by this pathway in 2 hours. But if you are more inclined to use imaging, there are very nice tandem reporters that can tell you, I mean, basically you can visualize as autophagosomes form, and you can also visualize as they clear up, because when they clear up, it's a reporter that contains mCherry and GFP. So as the autophagosome will be yellow because it have both reporters. But then as they fuse with lysosomes, since lysosomes are acidic, the GFP is lost. So then you end with this red dot that corresponds to autophagosomes that have been degraded by the lysosomal system. So it's very, very quantitative, and for cells in culture works very nice. So the field, I mean, we share them among many people, so I'm happy to share them with whoever is interested.
All right. Yeah, actually, Susanna is asking me if she could contact you. Susanna, I'll give you her email at the end of the webinar.
Yes, please.
So, all right. Unfortunately, we need to move on, and I'm gonna present in 1 second. Many things going on here. Yes. All right. Sorry. Yes, I would like to introduce our next speaker, and who is Dr. Albert Quintana, and he's an assistant professor at Seattle Children's Research Institute in Seattle, US. And he will talk about deciphering neuronal susceptibility to mitochondrial diseases. So we're running a little bit short in time. I'm gonna give you the voice. Just let me try to find you. Albert?
Hello, good morning
Yes.
from Seattle. Yeah.
How are you doing? I'm gonna
I'm
mute
good.
myself, and then you can start with your talk. Thank you very
Okay.
much. Thank you very much.
Thank you for the opportunity to present my work. Can you see my presentation? All right, let me know when you can see me. All right, can you see my presentation now?
Yes, please. You could start. Sorry.
Okay. All right. Sorry, I was
Go
waiting.
ahead. Yeah.
So yeah, thank you very much. All right. Yeah. So I'm very honored to be given this opportunity to present my work, and given that it's like really early now here in Seattle, I would like to start talking about energy, basically because everything that we do in our lives, like everyday activities like breaking a world record for some of us, taking an exam, or just attending to a seminar, even if it's on the web, it all requires energy. And we want to believe that we get that energy in different flavors, in different fashions. For instance, for me being here, moved from Spain, sunny Spain, to Seattle and the rain, just the idea of the sun and the beach is really what really charges my batteries back. But in case that's not present. I mean, everybody can think of coffee as a way to get some energy, or if you are really lazy, you can just go for a 5-hour substitute and get your energy back. However, if we think of scientists, I mean, we think that everything comes down to ATP. ATP is the base that all cells are going to use for energy. And if we think about ATP, we're thinking about mitochondria. So mitochondria is going to use the pyruvate that is going to be transformed from the glucose that we eat mostly, and it's going to get transported into the electron transport chain that has like these 5 complexes that it's going to produce the ATP for most of our cells to survive. And therefore, it's quite easy to see that if you don't have that energy, if that complex is not working, or if there are mutations in many different places of this system, you're going to develop like some serious deficits that is going to lead to cellular death, and in most of the cases, unfortunately, to early death of the patients. And this is considered, I mean, this is called primary mitochondrial disease. The interesting thing about mitochondria is, like, as you all are aware, it has its own DNA. So all these mitochondrial diseases are really complex because they can have like different components from nuclear or mitochondrial DNA and mutations. However, they are fairly, I mean, they are rare, but they are common in a way because they affect 1 in every 4,000 children that are born. And they all, even though they are really variable, as I said, they all have like common symptoms like myencephalitis, therefore affecting like tissues that require more energy than others. And they present hypotonia, ataxia, and as I said, most of them present death. The main problem is, like, as I said, there are many mutations. So therefore, studying mitochondrial disease, it's been like really complex because you have lack of tissue from human patients, and they are really variable. So in this case, we decided to make one of the few animal models for this disease using one subunit of the mitochondrial complex 1. Complex 1 is usually made of 45 subunits, so we just mutated one of them, NDAS4, that it's not part of the catalytic process of the complex 1, but it's supposed to be important for assembling or for keeping the complex together. So if you don't have NDAS4, and there are many ways, and as I said, there are many different mutations described in humans that can modify the activity of NDAS4 or its functionality. What it's believed is like the complex is really fragile and it can break apart and it doesn't assemble as well. And as I said, the problem with humans and problem with primary mitochondrial disease, it's like there's a huge clinical heterogeneity. So even patients with the same mutation can present like a really different phenotype. But we can all put them together and say that they have a mitochondrial disease before we can actually test which gene is mutated, because they have a failure to thrive, hypotonia, and usually brainstem or basal ganglia lesions. And unfortunately, in the case of this mutation, it's really severe, and the patients died before they were 2 years of age. So, as I said, we developed one of the few models for mitochondrial disease, and the first thing that we wanted to see is actually if they could recapitulate the human disease. And here you have a list of all the clinical signs that you see in humans, and interestingly, our mouse recapitulated all of them. So then the next question was like really, and it's like a pressing question, was like really trying to figure out why, where the pathology was coming from, what was the cause of this disease. And to address that, we generated a central nervous specific NDAS4 knockout. And in that animal, we could see that actually the main phenotype was maintained, suggesting that it was the brain, it was damaging the brain that was causing the phenotype of these animals. So then, if you look at the list of the different clinical signs, the second important question was exactly if we could identify what was actually killing those animals, that we could identify what's the region or what's the pathogenesis of the disease, and try to focus and narrow down our research. And if you look at all the clinical signs, the one that really caught our attention was like breathing irregularities, which are really present in humans. Humans with complex 1 mutations usually present breathing irregularities. So in this case, we decided to look at what was going on with the breathing pattern of these animals. And here you have a whole body plethysmography. So basically the animal is like sitting in a closed chamber where we can look at the differences in pressure, and we can correlate that as breathing of the animal. And if you look at the control animal, you can see that the breathing pattern is like really, really regular. Whereas in the knockout animals, we see like these huge apneas between bouts of respiration, suggesting that the breathing pattern of these animals was impaired. Furthermore, we could see that we could actually look at more parameters of breathing and respiration in these animals, and we saw that the minute ventilation, which is basically the amount of air that leaves the body of the animal per minute, was increased in the knockouts, both when they are really young and when they get old, suggesting, after looking at it, suggesting that it was actually that the volume was increased, although the frequency of the respiration was altered in those animals. And importantly, as the animal got older, and 50 is usually the latest stage of these animals, they don't live much longer than 50 days, you can see that there was a progression in the severity of the apneas, and the same for the irregularity of the breathing. So as the animal was getting older, their breathing was more irregular, and the apneas were longer and more frequent. And that's just breathing like normal air. If you challenge the animals and you present them to a low oxygen concentration or hypoxia, you can see that the knockouts presented like a lower response to hypoxia. They were not able to compensate as well as controls. And if you do kind of the opposite and present the animals in a high CO2 percentage, you can see that the knockouts were all affected, although we could identify like two different flavors, if we may. We had some knockouts that were really hyper responsive to CO2, whereas some of them were like really not responding at all, suggesting that the mechanisms to respond to high CO2 were altered in these animals. So the next question was looking, obviously we had seen that there was like a problem in breathing in these animals. We wanted to figure out if the respiratory centers were altered. So we did that, and by histological analysis, our surprise was that even though there were some difference at the level of the physiology of the respiratory centers, the main points of inflammation in the mouse brain of these animals were not in the respiratory centers, but rather they focus on the olfactory bulb, and then in different areas considered to be the vestibular system, so the vestibular nuclei, the inferior olive, the posterior cerebellum, and the deep cerebellar nuclei. And importantly, all these areas are connected in the vestibular system, which is basically thought as controlling or movement or balance. And as you say, it's affected when we are seasick, when we are motion sick. It's usually because there are mismatches between what your eyes are seeing, what your visual input is, and what your vestibular system is telling. So we also looked at the progression of the disease, and again, the only areas that really light up were the same that I just mentioned. You can see that as the animal gets older, the inflammation, we are looking here at microglial inflammation, microglial reactivity. You can see that it gets increased as the animal gets old and the disease progresses. In the olfactory bulb, in the cerebellum, some of the posterior lobes of the cerebellum are usually pretty much gone when the animal is like in late stage, and you can see that it starts to appear like symmetrical lesions in the vestibular nucleus, which are common for human mitochondrial disease, and then the inferior olive was also progressing. So then the question was like, we knew that there were like breathing deficits in these animals, but the anatomy or the histology of the damage didn't seem to correlate with those nuclei. So then we decided to think kind of out of the box and think if there could actually be like some kind of correlation or some connection between the vestibular system and breathing. And that was not, I mean, at first it looked weird because, as I said, I mean, everybody thinks of the vestibular system as balance. But if you think about it, the vestibular system does much more than that, and it has to do more than that. So as I said, the vestibular system is going to get all the input from the cranial nerve 8 from your basically your inner ear, and it's going to project to the vestibular nucleus, which is going to talk back to the deep cerebellar nuclei, the fastigial nucleus, which also talks back to the vestibular nucleus, and that projects to the inferior olive. And again, that is like a feedback loop. And basically, it's going to project to different parts of the brain and the spinal cord to control your balance, but in the sense that every time that you change your posture, every time you move, your balance has to correct for all the different forces, such as the gravity, that are affecting your body, and has to correct for all autonomic systems to keep basically everything normal, even though you're moving. So therefore, we were thinking that maybe the vestibular system, if it was affected, it was somehow affecting all those autonomic responses in different conditions where you are challenging the system. So to address that, we basically decided to create, with stereotactic surgery, just to create a knockout specifically in the vestibular nucleus. To do that, we injected a viral vector expressing CRE just in the vestibular nucleus. And when we saw, after we analyzed the animals, first of all, it's like there was like an increase in glial activation in the vestibular nucleus of the animals that were injected with CRE, but not in animals that were injected with a non-functional CRE, suggesting that there was damage going on, or that the vestibular nucleus was really susceptible to lacking that endosphore subunit in the vestibular nucleus. If we looked at the body weight of the animals, we observed that after a couple of weeks after the injection, the animals started to lose weight, and interestingly, they also presented like motor deficits that were present and were progressing after a month after the surgery. And more interestingly, some of the animals, we could see that the mortality of the animals lacking endosphore only in the vestibular nucleus was increased. So then the next question was like trying to know exactly what was going on with breathing on those animals. And what we saw is like, whereas the response to hypoxia was normal, the response to CO2, to high concentrations of CO2, was pretty much blunted in the knockouts, in the vestibular nucleus knockout, suggesting that the vestibular nucleus was really playing a role in sensing CO2 levels in the brain and compensating for that. And also, when we assess the breathing pattern of those animals, the animals that lacked endosphore in the vestibular nucleus presenting a higher irregularity in the breathing. So basically, we could see that if we removed endosphore only in the vestibular nucleus, we were affecting both motor system, but also respiration on those animals, and that was leading to a higher mortality. So then the proof of principle that it was really important for normal breathing was doing the converse experiment. And what we did is like get a complete knockout, endosphore knockout everywhere in the body, every single cell, and try to rescue the expression of endosphore in the vestibular nucleus. We did that, and what we saw here, you can see that's the usual lesion that you would see in the vestibular nucleus of a knockout animal, and the inflammation and the damage was reduced in animals that had rescued endosphore expression. As you can see, endosphore was fused to GFP, so you can get the signal. Apart from that, we could see that the animals had some delayed pathology. I have to say that to do these surgeries, the animals had already ongoing inflammation, so we could not do the experiment before the animal got sick. But we could get like a delay in the inflammation and in the pathology. And more importantly, we saw an extension or a significant extension in the lifespan of these animals, suggesting that the intervention that we were doing just in the vestibular nucleus was really important to prolong the life of these animals. When we look at the breathing, we could see that at least partially, if the challenge is not too strong, it was rescued both for high, low oxygen environments, and the same for high CO2 environments in these animals. So when you rescue endosphore just in the vestibular nucleus, you somewhat restore the breathing capabilities of the CO2 and hypoxia sensing capabilities of these animals. And importantly, that re-expression reduces breathing irregularity and gets it back to the levels of a wild-type animal. So then, having all these questions, the two main lines that my lab is interested now is basically, on the one hand, trying to figure out how the vestibular nucleus predicts these breathing alterations, which has not been described. So basically, we want to see what's the circuitry behind this, and using some optogenetic tools to really figure out the mechanism and which neuronal pathways are involved. And the second one, and it's the one that I want to talk a little bit about, is the one that we want to figure out, which is the mechanism that actually makes those vestibular nucleus neurons more sensitive to mitochondrial disease. And that's something that it's ongoing project in my lab. So, and that builds under a recent paper that we published, saying that actually if you provide these animals rapamycin, what you can see is that those animals present a better outcome in a motor test, like the rod rod. So the animal has to hang onto a rotating rod that keeps accelerating, and that kind of evaluates the motor performance. You can see that whereas the regular NDAS4 knockout presents a progressive worsening on the motor response, the animals that receive rapamycin pretty much maintain. Yeah.
Just be aware of the time. Be aware of the time.
Yeah. It's one slide, and I'm done.
All right. Carry on.
Sorry. Sorry. So basically, as I said, those animals rapamycin restore that effect on the motor control, and more importantly, daily administration of rapamycin prolong the lifespan of the animals like 5-fold. So, and importantly, it reduced the brainstem pathology. So we know that just blocking mTOR on those animals reduces the pathology and extends the lifespan of those animals. And basically, we still don't know the mechanism why that is happening, but as I said, we have observed that the animals have a baseline mTOR activity. So mTOR activity is baseline increase in those animals, and we believe the rapamycin is bringing down those mTOR levels in the animals. And just to say, just as a final reminder, I mean, mitochondrial function is not affected, so rapamycin is not acting directly on the mitochondria, and it's not due to the immunosuppressant effects of rapamycin, because another drug similar to rapamycin that does not block mTOR didn't have these effects. So just to finish, just mention the people in my lab and the different collaborations, and I'm open for questions. And as I said, we're trying to figure out which are the mechanisms of this mTOR function on lifespan expansion. Thank you.
All right, thank you. Thank you very much, Albert, for this talk. And if there's any questions, please raise your yellow hand or just write it down for me. Oh, there's one. Let's check. Yeah, there's one question. So, Jean-Jacques, I'm gonna give you voice, and you can formulate your question for Albert. There you are. Jean-Jacques, hello?
Yes, could you hear me?
Yes, we do.
Yeah, I can.
Hello, could you hear me?
Yeah.
Okay. I've understand that the mitochondria are all parts of your study, or in the center of your study. And my question is about when you look at morphology of the affected cells or affected tissue under the microscope or another tool of investigation. But do you see the change in? Do you see the change, a decrease or the increase of the number of the mitochondria compared to a normal cell?
So basically, we are looking at that right now. I mean, what we have seen is like there's differences in the ultrastructure of mitochondria. I mean, in affected cells, the cristae are collapsed, and we have believed that there's like an increased fission processes in mitochondria. So probably, I mean, the mitochondrial numbers, if you count them as, you know, total mitochondria, probably is going to be increased, but it's because there's more fission going on. And we're trying to figure out now with different systems really to label only mitochondria from those affected cells in the vestibular nucleus and try to compare and see what's really going on with the dynamics of those mitochondria. So yeah, it's something that it's ongoing now in the lab.
All right, thank you very much. I think we should carry on with the next speaker. So our next speaker, our third speaker is Dr. Francisco Molina Holgado, and he's reader in neuroscience as well as program convener of the Bachelor of Science in Biomedical Science at the University of Roehampton. His group has been working for at least 10 years on identifying and understanding the mechanisms and mediators of the neuroprotective actions of the endocannabinoids in the brain. So please, Francisco, I'm going to give you now the voice, and whenever you the voice as well as the screen. And whenever you are ready, you can start. Just give me 1 second. Yep, Francisco.
Okay, good evening from London.
How are you doing? Just wait a second. Yes, now your presentation is loaded, so you can start whenever you feel free.
Okay, thank you, Enrique. Thanks a lot. Thank you for inviting me to present our research here in this webinar. Thanks a lot. I got to speak. The title of my talk is "The Brain Cannabinoid System is Working for You." Basically, I got to tell you a couple of ideas about what is the role of the brain cannabinoid system, basically oriented to neuroprotection and to regulate neurogenesis. I would like to start with this slide, okay, that reflect what is the highly dynamic situation that occur in our brains, okay. The brain is capable of structural and functional reorganization depending on a variety of events. Here in the left side of this slide, you have three key levels of activity in the brain: maintenance, protection, and repair. Maintenance of the homeostatic environment, protection, and repair regeneration. Okay? And here in the right side, you have key points related to maintenance: brain homeostatic network, neuronal turnover, synaptic scaling; protection, synaptic and neuronal integrity; and related to the repair regeneration process, basically synaptic plasticity and adult neurogenesis. In the case that any of these three levels of functionality is not working properly, appear the neurodegeneration process. Okay, basically in neurodegeneration, the structure to protect is in this picture that you have here. This is a scheme about synapses. And this is the key structure that is necessary to maintain for the proper brain functioning, or for the proper, or to maintain the homeostatic environment of the brain. In the last 15 years, we was working with different natural or synthetic cannabinoids to try to maintain this structure in different processes of neurodegeneration: multiple sclerosis, Alzheimer, motor neuron disease, Huntington disease, basically in any kind of pathology that goes to alterate this synaptic contact. Okay? Endocannabinoids released by our brains, all the mammals we produce endocannabinoids in the brain, maintain this structure, are neuroprotective, if you like. You have here in the right side a book edited by the British Medical Association several years ago about the therapeutic use of cannabis. Our research, our lab is interested in the study of these endocannabinoids in terms of neuroprotective activity and neurogenesis. To give you an idea of the relevance of the use of cannabinoids in clinical trials, I take this morning this picture for the clinical trials database for the National Institutes of Health in USA, and you can see here at this moment we have over 100 clinical trials that are using cannabinoids in different neurodegenerative diseases. This is the first page, but you have here schizophrenia, Huntington disease, multiple sclerosis, but we can continue with the list of neurodegeneration. This means exists a real interest in the clinical for the investigation of these compounds as potential neuroprotective agents. Okay. We are speaking about cannabinoids, but what is the brain endocannabinoid system? Okay, basically it's a neuromodulatory system comprising the endogenous ligand that we call endocannabinoids that are produced for our brain. Basically are derivatives of the arachidonic acid 2-AG, 2-arachidonyl glycerol, and anandamide. We have these endocannabinoids, and these endocannabinoids go to bind to a specific cannabinoid receptor, CB1 and CB2. In the past, we were thinking that the proper brain cannabinoid receptor was the CB1 and the CB2 was only present in the immune system. But today we know that both CB1, CB2, and probably as potential CB3 are present in the brains. This means the brain endocannabinoid system is comprising these endocannabinoids, these cannabinoid receptors, CB1 and CB2, and all the enzymatic machinery that is necessary for the regulation of the biosynthesis and degradation of the ligands. You have here a couple of these cartoons represent what is the CB1 here in my left side of the slide. The CB2 are typical G protein coupled receptor with these 7 transmembrane fragments. Okay, this means the presence of cannabinoids, the distribution of the endocannabinoid system, it is a constant in all the mammals. Here you have a sagittal section of a rat brain, and you can see different areas that the cannabinoid receptor are present: cerebellum, brainstem, substantia nigra, and basal ganglia and hippocampus. This is in rodents, but what occurs in humans? You have here in humans the 3 areas in the left side of the slide. The 3 areas with the highest expression of cannabinoid receptor are the brain, the digestive tract, and the reproductive system. And here in the right side you have PET and magnetic resonance. Intensity of color means more color that you have here, red, you have in this part, highest expression of cannabinoid receptor. This means we have all this machinery in our brains. Interestingly, we have this machinery in our brains, and I would like to mention that the most abundant receptor in our brain is the CB1 cannabinoid receptor. This receptor is more abundant than all the dopamine, noradrenaline, serotonin, and 10 times more abundant than the opioid receptors. This means this highest expression of this maximal expression of this receptor, we can use this system. Okay. You have here, because as you know, is controversial, the use of psychoactive cannabinoids in therapy in clinica. Now we know that natural cannabinoids that we call phytocannabinoids that are produced by the cannabis sativa plants, non-psychotropic cannabinoids, they have a widely use that you can have here. Different areas. I am very interested in all the possible or potential effects in the brain from antipsychotic drugs, anti-epileptic, neuroprotection. These drugs are anti-inflammatory and immunosuppressive. And you can see here the potential use of these phytocannabinoids, and I remark are non-psychoactive, because this was a limitation in the past to use this drug, is absolutely fantastic. What is the functioning of the role of this brain cannabinoid system? Several years ago, with my collaborators in Spain, we proposed that the endocannabinoid system is a functional interface between the brain and the immune system. And of course, what to control, as I mentioned before, neuroprotection and neurogenesis. What means this? The immune system and the brain both express all this cannabinoid machinery. This means cannabinoid signal in the immune system communicate with the brain, and this is a bidirectional communication. And this is relevant because this is very important in terms of neuroprotection. Think in terms of neuroinflammation, all the regulation of the pro-inflammatory cascade with cytokines, mainly interleukin-1, tumor necrosis factor, and now we know that this connection is very important in neurogenesis. This means neuroprotection and neurogenesis, they have a common substrate that is the immune system. This means it's necessary inflammation because it's necessary repair, and these inflammatory mediators are key in neurogenesis. You have here our first experiments when we identify downstream activation of the cannabinoid receptor, the cytokines that regulate this cannabinoid receptor. Basically, the key cytokine that this cannabinoid receptor regulate is the interleukin-1 receptor antagonist. This is a natural antagonist for the actions of the interleukin-1 beta in the brain. Interleukin-1 beta is a pro-inflammatory cytokine. You have here what is the how we describe the release of the transcription of the interleukin-1 receptor antagonist via cannabinoid activation. And this is key because this cytokine, interleukin-1 receptor antagonist, go to block several inflammatory processes in the brain. At the same time, and I mentioned now, I go to explain this a little bit later, interleukin-1 receptor antagonist is key neurogenesis. That related to neuroprotection, but what occurs about neurogenesis? Okay, we have demonstrated in vivo and in vitro that the activation of the cannabinoid system go to promote neural stem cell proliferation. In the right side, you have the models that we use with neurospheres that they use that they express CB1, CB2 cannabinoid receptors. They are SOX2 positive and nestin positive. This means these are proper neural stem cells, and the fate of these cells is depending on the CB1, CB2 activation. In the right side, you have pictures from a publication with my college at King's College, where using a tracking system, we track the progenitors, the neuroblasts that migrate from the subventricular zone to the olfactory valve that you have here. Activation of the cannabinoid system in the subventricular zone go to produce an increase in the proliferation, in the appearance of these neuroblasts in the olfactory valve, and these progenitors go to integrate in all the circuitry in the olfactory valve that you have here. What occurs if we block the production of endocannabinoids? Basically, this is in the micrograph B, if we block the production of endocannabinoids using a blocker of the diacylglycerol lipase that go to convert diacylglycerol into arachidonyl glycerol, we have a reduced migration of these neuroblasts. This means the brain cannabinoid system regulates in vivo or in vitro the production of new neurons. I mentioned a connection with the immune system via cannabinoid activation. You have here, this is a recent review that we published in journal Neurochem, how the activation with exogenous, with synthetic cannabinoids or with endogenous cannabinoids ligands, the CB1 receptor, the CB2, and this other receptor that I mentioned as a potential CB3 that we call GPR55, downstream of the after downstream activation of this receptor, there are different pathways that are active. Basically, protein kinase B, AKT, ERK, STAT3, that is the key machinery for the interleukin-6 family, like the GP113. What occurs with this? There are different mediators that are increased in neurogenesis, like tumor necrosis factor, or interleukin-1, interleukin-6. What occurs in our experimental models of neurogenesis if tumor necrosis factor is increased? Okay, we go to observe a neural stem cell proliferation. In the contrary, if the cytokines that are increased are interleukin-6 or interleukin-1, we go to observe a big process of differentiation of these neural stem cells. We have confirmed all these experiments with transgenic animals for interleukin-1 and for interleukin-1 beta knockout mice and tumor necrosis factor knockout mice. Recently, we have developed a new approach to try to understand all this situation, how cannabinoids regulate neurogenesis, and using microarrays, we have investigate what are the genes, the pathways, and the networks that are controlled by this brain cannabinoid system. You have here, this is unpublished results from our last experiments with microarrays. What are the key networks that the brain cannabinoid system control? I would like that you focus here, number 1, 2, and 3. Basically, the first network is the development of neurogenesis in general, and the networks 2 and 3 are related to inflammation. This means we demonstrate that the brain cannabinoid system is neuroprotective and at the same time regulate neurogenesis. It's an inducer of neurogenesis. Finally, you would like more information about our experiments, you have here my contact details, and you can send me a message or use my or contact me via Twitter. Thank you very much for your attention.
Thank you very much, Francisco. So, I didn't know about this, but it seems like through these talks there is a quite interplay between inflammation and regeneration, which is exactly the same that is happening in the heart. I'm just got curious about that. And my question is, when you show the picture of the receptors, you show that the receptors are mainly in the brain, digestive tract, and the bladder. Do you have any idea why the receptors are located in these three particular organs? I just got impressed by this picture.
Okay, that's a very good point. We know, for example, that there is a big connection between the liver and the brain, okay? If you modify inflammation in the liver, this is going to have a consequence in the brain. And we perform different experiments in animals that we regulate brain inflammation via actions in the intervention in liver. This means to me is a clear connection between the liver and the brain, or the digestive system and the brain. I don't know what is the key in the reproductive system.
All right, good to know. We will start using cannabinoids anyway. Is there any more questions around? If no, I would rather go to the next talk. Just give me 1 second. Thank you very much, Francisco.
Thank you.
All right. So, yeah, thank you very much, all of you. And as a concluding remark, it's a shame that Raul couldn't make it through. It couldn't be even more interesting with him being around. I hope everyone enjoyed. And I would like to point out, perhaps, that this will be the first but not the last neuro webinar. We need to convince Raul to make another one. And the second thing is, I think these are really hot topics, and we have a clear example with the passed away of Adolfo Suarez 2 days ago. So I think through these talks we can, I don't know, send this message to the general public that although there's a lot of work already done, there is a long way to go. And finally, I'm showing you here the list of the cycles of webinars. And if there anyone in the public that wishes to present a webinar, please send me an email. You'll see it in the next slide. And let me tell you what we have so far. We have a slot free in May, so feel free. Then we have another webinar on July 2, 2014 on nanotechnology. Another one on management and restoration of contaminated land and groundwater on September 2014. I'm trying to convince people to make a cardio webinar just to fight with the neuro webinar people, and this will be around 2014. And finally, at the end of 2014, beginning of 2015, I'll try to bring both the elected presidents of both societies to give us another idea of what happened the year before and what are the challenges for 2015. And finally, I would like to thank again all the invited speakers for this formidable webinar, and thank you all for attending to this fifth webinar of both societies, TERFA, SRUK, THERU. Again, if you wish to contact us or to wish to contact any of the speakers, just drop me an email to any of these two email addresses, and I will be more than happy to put you in contact. Thank you very much, and thank you all of you for attending. Oh, here it is.